just summarize everything in 5 minutes, okay, whatever we have discussed previously. Correct na? Blood is what kind of a tissue? Fluid connective tissue. Correct na? Ultimate function of the blood is what? Transport. Correct na? Transport. Transport is the major function. What is the major temperature of the blood? Temperature, not the pH. I told you now, see, body temperature is 37.5 degree. The blood is around 38 degrees. Correct na? That is the temperature managed by this particular what we can say blood. Correct na? And what else we have started discussing later? Later we understand blood is low viscous or high viscous. Which is extremely high viscous in the blood, water, oil? Oil is high viscous, followed by blood, followed by the water. Correct na? I'll quickly summarize this point very effectively. When we consider the classification of the blood, what we understand, friends? So blood, how much percentage of the body weight composes the blood? 8%. Correct na? If you take a 70 kgs healthy human being, 8% of the total body weight composed by blood. If I take this entire blood as a 100%, 55% of the blood is plasma, 45 is formed elements. Correct na? If I take 55% of the blood plasma as the 100%, what I will get, friends? So mostly it is made up of water and proteins. So I believe everyone remember we have albumin, globulin, fibrinogen. Among these, which is most abundant? Albumin. Abundant in the sense percentage. Okay. And very important question. These three proteins are released by which cells? Name the cells which releases these three. Correct. What are those? Hepatocytes, which are liver cells. Please do not confuse with the statement. Liver cells are generally going to release them. You're getting the point. But they are entering into where? Plasma. Correct na? Now very simple question. Among these three, which is not involved in the transport? Which is fibrinogen. Fibrinogen is exclusively involved for what function? Clotting. Correct na? We have how many globulins? Alpha globulin, beta globulin, even gamma globulins also there. Some involved in the transport, some is involved in the immune system. What is the function of albumin? Osmoregulation and? Very good. Transport of what? Transport of lipids. Correct na? We get energy from fatty acids. Correct na? Where the fatty acids break down occur? In the adipose tissue. So which means fatty acids has to be transported into the adipose tissue via blood. In the blood, suppose example, this is a blood vessel. Inside the blood vessel we have albumin. Inside the albumin we have fatty acid. So do not confuse. Albumin is a protein, but it is acting as a transporter. Transporter of what? Lipid. That's why it is called lipid transport. Correct na? And remaining is how much? 45% RBC, WBC. How much percent RBC? 90. Very good. 90, more than 99%. Only less than 1% we have WBC and platelet. If I do the process of the centrifugation, which will settle in the middle? WBC and platelets, because they are having lower density compared to the RBC and more density compared to the plasma. Plasma settled on the top. Correct na? Because plasma is what? Plasma is completely water. Water means a liquid which is keep on floating on the surface. Correct na? Good. So later we understand very clearly the major functions. Correct na? Now don't get confused, friends. What is the total lifespan of RBC we studied? 120 days. That is what we actually studied. Correct na? And a very, very important statement I want all of you to understand. What is the name of the cell in, relax, what is the name of the cell in which RBC destruction happen? You're saying something, what is that? Spleen is a cell? Spleen is a cell. It is what? Organ. I am asking what is the name of the cell, which is macrophage. Correct na? Macrophages are the cells which are present in the spleen and liver, where RBC destruction occur. So if they ask you a question, which organelles are said to be the graveyards of RBC? Spleen. But if they ask you a question which cells are acting as a graveyards of RBC is macrophage. Now tell me, RBC having mitochondria? No. RBC having nucleus? No. But I believe you all remember when RBC is newly formed, it is having nucleus. We call pro-erythroblast. Upon maturation, we call it as reticulocyte. Then reticulocyte eject the nucleus. When the reticulocyte enter into the blood circulation, we call RBC. Correct, na? Now the most important one, RBC, what it is not having what? Mitochondria and nucleus. Nucleus also, followed by Golgi complex. If it is not having mitochondria, it cannot perform cellular respiration. What's the ultimate function of RBC we have studied? It is involved in the transport of oxygen. As you all people know very well, if it is involved in the transport of the oxygen, anything which is mediated with transport is energy consuming. So if the hemoglobin wants to be transported, we require a lot of energy. But what we discussed, RBC do not have. Hence, it cannot produce ATP aerobically. Then how the RBC produce the ATP? Anaerobic respiration. This is the question asked in the exam. See, the question is crystal clear. The function of RBC is transporting oxygen. RBC never ever use the oxygen whichever it is transporting. It never use. RBC always perform anaerobic respiration and produce little amount of ATP. By using that ATP, it can transport oxygen. Okay, you people watch the previous class, by that you get a good idea. Okay, both of you. Good. So I think this is what we were discussing. Okay, let's continue. So they are broadly categorized into what? WBCR, granulocytes and agranulocytes. Why do we call granulocytes? When we observe under light microscope, if the cytoplasm contains conspicuous granules. Conspicuous means some crystal-like granules. We call granulocyte. And if we did not find any granules in the cytoplasm, we called agranulocytes. So the best example which we already studied, friends, so you can see very clearly, right? The cytoplasm is full of granules. You can see that, right? Here the cytoplasm is do not contain any kind of a granules. That's why they called agranulocytes. Agranulocytes are differentiated into lympho and mono. What's the simple difference between monocyte and macrophage? Whenever it is circulating completely in the blood circulation, we call monocyte. So which suppose all of a sudden a pathogen entered, where pathogen entered in the liver, then automatically monocyte enter into the liver tissue. Whenever it enter into the liver tissue, we don't call it as monocyte, we call it as macrophage. Since it entered into the liver, it is having a different name. Okay, that we will read in the immunology. We call Kupffer cells. Correct, na? So it is basically a macrophage only, but it enter into the liver, we calling it as Kupffer. Suppose it enter into the kidney, we call glial cells. Okay, that we will read it separately. Okay, depending on what tissue it is entering, it is having different type of name. No point of confusing there. And most importantly, lymphocytes are further divided into B, T, and what we discussed, friends, B lymphocytes further divided into plasma cell and memory cell. Plasma cell produce antibodies. Memory cell store the copy of antibodies. Correct, na? Now what happened? You're fine. Okay. Next, T lymphocytes further divided into T helper cell, T cytotoxic cell. Anyway, we all read this in the immunology chapter, human health and diseases. T helper cells, they are the one which help the B lymphocyte to produce antibody. We'll read over there. 99.99% all the HIV virus act on T helper cell. That's why our immune system will become compromised. And what's the function of T cytotoxic T cell? Everyone remembered. Cytotoxic T cell and natural killer cell works on infected tumor cells or virus infected cell. This is a normal cell, healthy cell. It is infected by virus. Now I call it as virus infected cell. This virus infected cell can be destructed by natural killer cell and a cytotoxic T cell. These all belongs to the lymphocytes. Correct, na? Now let's quickly understand. Where is this we have mentioned? Okay. have updated a PPT, why it is not coming? Cha. Okay, give me 2 minutes. I'll bring updated PPT because here less information is there. Huh? Just 2 minutes of time. Sometimes brain won't work. All right. See, total human physiology, there are around 1200 slides are there. Even if you read these 1200 slides instead of the modules, you get a lot of information. By that you can answer any kind of a question, that is coming in the exam, especially if the question is too tough. Perfect. Because we have to prepare in such a way that the question should not go beyond the level whatever we read. Even the question comes extremely difficult also, we should be in a position to answer this. Okay, all right. Okay. So granulocytes are what? Neutrophils, basophils and eosinophils. Whenever we are reading the neutrophils, what is the first statement all of you has to remember? First statement, remember, very good. Neutrophils are involved in the process of phagocytosis, which means all of you remember neutrophils are the cells that enter into the damaged site first. So which we called early responders. Okay, suppose example, there is a small cut happen. What happens generally? Lot of blood will leak or blood will flow. So through all those blood vessels, abundant amount of neutrophils entered into the damaged area and start the process of phagocytosis. That's why neutrophils are said to be the early responders to the process of inflammation. So now I want you to remember compulsory the extra points which are not there in the NCERT and modules. Of course, neutrophils contain granules, that's why we call it as granulocytes. And very interesting point, whatever the granules that are present in the neutrophils, all the granules are extremely smaller. And very interesting point, even though they are smaller, they are distributed evenly. Okay, suppose now look into this, this is the granulocyte, it is having abundant amount of the granules, all the granules are distributed evenly and extremely smaller. Now neutrophil, whenever it is in the young stage, okay, we called young neutrophil and we called mature neutrophil. So please don't confuse with the name. Whenever the neutrophil is young, we call it as band. The name of the young neutrophil is called band. Whenever it is young, the nucleus is bilobed, just like this, that's all, only having 2 lobes. But whenever it become mature, we call it as a multi-lobed, correct na? We call multi-lobed. That multi-lobed only we call it as the polymorphs or polymorphs or polyp. This is the question. Okay, so in simply called, friends, PMN, polymorphonuclear leukocyte. Okay, simply remember polyp. So what's the first point you have to remember? Neutrophils plays a key role in the phagocytosis. Second point, neutrophils are said to be the early responders to the infection. The third point, young neutrophils are called bands, they are only bilobed. Mature neutrophils are called as? Very good. In simply you remember polymorphs or polyps, polymorphonuclear leukocyte. Correct na? That's the only thing you have to remember them. Now friends, when we look into the eosinophil, what I told you about the eosinophils? Remember? Very good. Eosinophils are the type of granulocytes that plays a key role against parasitic infections. So we have a lot of worms, okay, so that worms can be destructed easily by eosinophils. Okay, take good rest. Okay, so of course they are also granular leukocyte. Just now what we discussed in the neutrophil, all the granules are small but they are uniformly arranged, correct na? But when we look into the eosinophils, friends, can you see that all the granules are larger? Even though they are larger, they are also they are uniform and evenly distributed. Don't get confused. Now can you see this? This is nucleus is how many lobed? Bilobed, correct na friends? So it is just a bilobed. Okay, now very important statement, we are using word bilobed, which means 2 lobes are there. Or we use the word multi-lobe, means multiple lobes are there. Now question here, how one lobe can be connected with other lobe? That is important. One lobe can be connected with another lobe with a thick structure called as chromatin band. So remember the point correctly, it's not there in NCERT. Chromatin band is the name of the structure which connects the 2 lobes, whether it's a 2 lobe, 3 lobe or multi-lobe. So what's the first point you have to remember? Eosinophils plays a key role in parasitic infection. It is large granules, uniform sized, correct na? Now the nucleus of eosinophil is mostly 2 lobes connected by thick strand of chromatin, and the most important for the exam is basophil. The moment you heard the name of a basophil, so remember, first point you have to remember, basophils are the one which plays a key role in allergic reactions or inflammation. I will tell you in the chapter of immunology to all of you very briefly, but remember here, most of the basophils and most of the mast cells release same chemicals. Okay, and what are the chemicals released by the basophils? Tell me. Very good. What is the function of histamine? No. It is not anti-inflammatory. It causes the inflammation. It constricts bronchi. Okay. I told you one example, right? When we go to a green park, we have a lot of pollen grains. So people start all of a sudden sneezing, running nose, eyes become reddish. Why? Pollen grains are acting like allergens. Allergens means which cause the allergy. They enter into the bronchi. They stimulate the basophil, release of histamine. Histamine come and constrict the bronchi. Then we feel extreme difficulty in breathing. After some time we feel relaxed. If the symptoms are persistent, what we take? Antihistamine, which suppress the level of histamine. Best example, what is that? Levocetirizine, which we generally take in our daily life. Correct, na? So basophil release histamine and heparin. What is heparin? Natural anticoagulant. Now we all read rDNA technology, na? What is the artificial anticoagulant which is developed by using rDNA technology? Okay, anyway, we'll read that. You have to remind me that on that day. Hirudin. Okay, which is developed by using rDNA technology. Okay, we're gonna read it. But works like a same heparin. And what else it contains? Serotonin. What is the function of serotonin? Very good. What is that? Vasodilator, and serotonin is a powerful neurotransmitter. Correct, na? It is also released from basophil. And PAF, platelet activated factor, which is responsible for the blood clot. See, these are all not there in NCERT or modules. You have to remember mandatorily because the questions were asked in the exam. Okay, so basophils are granular leukocyte. Now friends, compared to the neutrophils and eosinophils, the granules are very larger only in the basophil, very thick and larger granules. Why? Because the granule stores all these chemicals. Okay, neutrophil is multilobed, eosinophil is bilobed, basophil is also bilobed. So the only granulocyte which is multilobed is neutrophil. No point of getting confused over there. Okay, so these are all like granular leukocytes. We're gonna read them in the human health and diseases under immunology. Okay, we no need to read over here. The next one is platelet. Okay, tell me Kusma, what is the name of the growth factor that is required for the process of erythropoiesis? What is erythropoiesis, Sanjana? Formation of RBC. So tell me Ayesha, we require a growth factor which stimulates this process. What is that? Erythropoietin. Who will release this? Kidney. Correct, na? Juxtaglomerular cells of the kidney. So what we discussed, if there is a kidney failure, RBC production also never happen, which means no hemoglobin, no oxygen. Obviously, it eventually leads to death also. Very dangerous condition. Similarly, however, erythropoietin is required for the production of RBC, we also require okay, for the production of platelets. This thrombopoietin is released from liver. So if I have a liver impairment, blood clotting also never happen. You're getting the point. I have a liver impairment, but even the small injury occur, it takes an hour sometimes for the blood to clot. In general, blood clotting occur in 6 to 7 minutes only. You might observe in some diabetic patients, blood clotting occur after 10-15 minutes. Sometimes it will take hour also. Correct, na? Because of the impairment of thrombopoietin. Can you see, friends? I believe you remembered. First, RBC produced as a pro-erythroblast. After that, reticulocyte. Then, red blood cell. Similarly, can you see, friends? We call first hemocytoblast. What is that? Hemocytoblast. Which is the stem cell. Hemocytoblast converted into megakaryoblast. Why we call megakaryo? Mega, large. Karyo, nucleus. The blast cell, the cell which is having larger nucleus, that's why we can call megakaryoblast. Megakaryoblast differentiate into pro-megakaryocyte. Pro means always immature. Correct, na? We use the word pro-insulin. Why? Because it is immature insulin. Correct na? Similarly, we use the word pro-megakaryocyte. Pro-megakaryocyte differentiated into mega. Then finally, there is a release of platelets. Correct na? Hardly we have only 2000 to 3000 platelets only. Very less in number only we can say that. Now friends, very important. I believe you all remember, now answer all of you. Significant reduction of number of RBC, call it as? Very good. We called as anemia. Increasing in the RBC named as? You are not reading. Huh? Polycythemia. Once again, remember my statements. It is not important how much time we read. It is only important how much you remember. Focus on remembering rather than reading. Correct na? Because this we can get in the matching question, and which we already got in the exam. Anemia is low number of RBC. Polycythemia means high number of RBC. Anyway, both are dangerous condition. Anyone remember why, when or which condition RBC number becomes more? Anyone remember? I used a word, tissue hypoxia. What is called hypoxia? Low oxygen. Especially when we are climbing a mountain. Then what happens? Oxygen level become lower. Then kidney stimulates, release erythropoietin. Then polycythemia occur. Correct na? See, biology is always linking. The more you link, you can remember easily. So friends, platelets are also called as thrombocytes or cell fragments. Why did we use the word cell fragment? Because in the previous picture, can you see the picture now? Megakaryocyte blast and given rise to lot of fragments, small pieces. That's why we use the word cell fragments. Produced from the megakaryocyte in the bone marrow. Correct na? Now very important, they are round, irregular, biconvex and nucleated. That is more important, which you have to. RBC do not have nucleus, whereas WBC having nucleus. We have granulocytes, all are nucleated only. Platelets are also nucleated. The formation of RBC called as erythropoiesis. The formation of thrombocyte called as lysis. Okay, don't get confused over there. Of course, production taking place in the bone marrow. So if RBC number got decreased, we call RBC number decreased, anemia. Platelet number decreased, thrombocytopenia. Increased, cytosis. Okay, always remember SIS, cis, always talking about production. Okay, anemia, always talking about decrease. I'll ask you the same question in the next class. Cytosis is increasing in number, cytopenia is decreasing in the number. No confusion over there. Okay, now after the blood cells, majorly we have to focus on what? Blood groups. See, right now whatever I have explained is a small introduction. Let's consider in this entire chapter we discussed just 1%. Okay, so the real lesson starts from blood groups, because I already told you in the previous year exam we got the questions either from blood groups or from coagulation of the blood or from functioning of the heart or from ECG or from cardiac control or cardiac output. Only these 5 areas where the previous questions were appeared. So focus. Blood groups. I believe you read the genetics in your second year. Okay, you people read the genetics? Not yet. Anyway, madam will teach. I will also teach one more time. Blood groups is a best example of multiple alleles. Correct na? Yes. Generally one gene having 2 alleles. Okay, but this is the best example for multiple allelism. Correct na? So now if I look up blood, in all the person it appears to be red in color. Then what we generally think, everyone having this similar type of a blood externally. But when I see internally, the blood will be completely different from one person to the another person. So how it is different from one person to the another person? It is exclusively based on their chemical differences, exclusively based on their antigens. Correct na? Tell me, how can you give the definition of an antigen? Okay, all of you answer my simple question. There is pathogen, okay? There is antigen and there is antibody. What is the difference? What is pathogen? See, any foreign particle or any microorganism having the capability to cause the disease. Which cause the disease is? Pathogen. Now the question, a bacteria enter in my body. How it cause the disease? By releasing antigen. So the foreign particle, foreign substance released by the pathogen in the body which cause the disease is antigen. Suppose you take an example, HIV virus. How the HIV virus attack on the cell? What it release? HIV virus release RNA. Suppose a bacteria enter in my body. How bacteria cause the disease? In the entire bacterial body, which is the strongest structure? Cell wall. So it may release its cell wall components. Bacteria it may release its DNA. Bacteria release its flagella. Anything released by the foreign organism is called as antigens. Our body has to destruct this antigen. So it release antibodies. Okay, antibodies are proteins which destruct the antigen. So based on that antigen, we have categorized the blood group into ABO system. Okay, I'll explain slowly, everyone focus. Even though you read 100 times, there is no use. If you understand once enough, you can answer every single question. ABO blood group system was discovered by a Karl Landsteiner, because in the NEET exam, out of 90 questions, at least 1 or 2 questions comes from the name of the scientist. So it is idealized to read the name of the scientist. Okay, so what he discovered? ABO blood group. And we're going to discuss elaboratively. It is also include something called as RH group. Not every individual having RH group, only some. The name RH came from rhesus monkey, in which it is identified. The most common is ABO. So according to the ABO blood grouping system, persons having the A group, B group, AB and O blood group. Correct na? Now let's understand it very clearly. Now friends, how did I categorize into A blood group, B blood group and AB blood group and O blood group? Okay, look into the picture, then we will read line by line elaboratively. This is what? A blood group. When I can say a person having the blood group A, this is what cell? RBC, correct na? RBC having membrane, correct na? Whenever the membrane of the RBC, if it contain an antigen, what is that antigen? A antigen, then that blood group is considered as blood group A. Depending on what? Depending on the type of antigen present on the membrane. So it is blood group A, correct na? So blood group A contain A antigen. Undoubtedly RBC also contain antibodies, correct na? Antibodies, where it is located? Now tell me all of you. In the plasma, correct na? So in the plasma we have antibodies. Blood group A having an antibody called as anti-B. Anti against. Against what? B, not against A. If it is against A, that own cell will be destructed. You are getting the point. So blood group A antibody is B. Anti-B. No, it's not a B. Anti-B. Against B. Against B. Anti means against. Against B antigen. But RBC having A antigen, then there is no problem. Now when we can call blood group is B? When on the membrane of RBC it is having B antigen. And in the plasma, anti-A. You're getting the point? Okay, now you tell me type someone is having AB blood group, which means so A antigen also present on the membrane of RBC and now what kind of antibody it contain? Because if it contain either anti-A or anti-B, it will destruct both. Then the RBC will be destructed. Hence, no neither antibody, neither anti-A or anti-B. Correct now, because we have both antigens. Now type O. When we can say blood group is O, it do not have A antigen or B antigen. But what kind of antibody it is having? anti-A antibodies as well as the anti-B antibodies. So both antibodies are there. So they may attack on A also, they may attack on B also, but there is no antigen. Correct, na? So now look into this. Due to the presence of antibodies, the person blood group A, okay, one second, this first we will read here quickly. One second. Okay, so people whose RBC display only antigen A have type A blood, while those who have only antigen B are type B. Individuals who have both A and B antigens are of type AB. Who have neither antigen A or B, they are type O. Sometimes in the exam, in order to make you confuse, they'll give a different words instead of using antigen and antibody. So antibodies, friends, it is commonly called as agglutinins. Okay, agglutinins. Okay, and antigens generally called as agglutinogen. Okay, you can easily remember antigen, agglutinogen, antibody and agglutinin. There is no point of getting confused over there. Okay, now focus. ABO grouping is based on the presence or absence of two surface antigens. Correct, na? On the RBC named as A and B. See, what is the question for the exam? This is the question for the exam. So antigens are chemically off. That's the question. They are, you all read biomolecules, right? What are glycoproteins? Now we read the protein classification, na? Simple proteins, compound proteins and derived protein. What is called compound protein? Compound or complex protein. It contains protein part also and non-protein part also. Remember or not? So if non-protein part is sugar, we call antigen is also glycoprotein, and we'll read in the human health and diseases. Antibody is also glycoprotein only, made up of sugar plus protein. Please don't forget this. Similarly, the plasma of different individuals contain two natural antibodies. They are also type of glycoproteins only. Antigen is a glycoprotein, antibody is also a glycoprotein. Now look into the interesting line. Blood types remain constant throughout lifetime. Correct or not? When I took birth, my blood group is A. It never changes during the time of during the lifetime. That's why they are constant throughout and they are hereditary. Now please remember, this is also a question asked in the previous year exam. Which gene is coding for these antigens or the glycoproteins? Which is gene 1, or you can call gene I also. And just now we discussed, right, blood groups are generally what? Multiple allelism. So gene I has 3 alleles. One is IA, IB and IO, or simply called I. You know what does I represents? Isoglutenin. Correct, na? That's what we discussed, right? Agglutinin, antibody, agglutinogen, antigen. This is called isoagglutinin. Okay, or simply remember as I. Correct, na? Now where generally serious reactions occur, the serious reactions only occur during the blood transfusion. During the time of the blood transfusion, it is very important that donor blood group and recipient blood group has to be matched compulsory. Suppose, listen, I will give you one very good example where tough questions can come. If the blood from non-compatible groups is used for transfusion, it may lead to clumping and destruction of RBC. Tell me, friends, what happened if the destruction of RBC releases? Destruction of RBC occurred. What happened? RBC contain what? Hemoglobin. Hemoglobin releases. That hemoglobin will go and block the kidney. Especially it will go and block the juxtaglomerular apparatus of the kidney, which leads to kidney failure also, which is very dangerous condition. So in a simple words, when wrong transfusion occur, RBC will break down. Then there is a release of hemoglobin that causes kidney failure in general. Now the anti-A antibodies bind to A surface antigen and anti-B antibodies bind to B surface antigen. Okay, now look into this very quickly because I have written line by line, by that you can understand it very clearly. Okay. Now focus. Similarly, person with blood group B can receive blood only of blood group B, correct na? Because blood group has to match. Whereas it can also receive the blood from O. Why? Because O do not have antigens, correct na? That's why O is said to be the universal donor. Similarly, if the blood group is A, he can receive the blood from A and O, correct na? But tell me, if the person is AB blood group, very good. So AB can receive from all. Why? Because AB contain A antigen also, B antigen also, correct na? And moreover no antibodies. That's why it is said to be universal recipient. One is donor and one is recipient. Since there are no antigens on RBC of a person with blood group O, and both anti-A and anti-B, the antibodies are present in the blood plasma, then people with O blood group can receive the blood group only from O, correct or not? So O can donate to all, but O can receive from only O. So don't confuse with that point. Anyway, we have written here, I want every one of you to remember this mandatorily. Okay, 1 second. Okay. So I want you to memorize this. There is no point of memorizing, you can easily understand. Blood type is A positive. E can give to whom? Donate to A. And you also donate to AB plus. AB is common because it is universal recipient, correct na? So now my blood group is A. Okay. From whom I can receive now? I can receive from A only, right? And O, correct na? Because O is the donor. So obviously O can give to anyone. At the same time my blood group is A, that's why I have to receive only from A. Correct na? A. But I can donate to A also and AB plus also. AB plus can receive from anyone. Then when it comes to the O positive, very good. It can donate to everyone. So it can donate to O also. It can donate to A also, B also, AB plus also. Correct na? Now I am O. From whom I can receive? Only from O. Simple. Then the blood group is B. Then I can give to B and AB. Receiving compulsory from O and compulsory from B. Now AB, which is, now don't get confused. AB gives to whom? Very good. But receives from everyone, since it is a universal recipient. There is no point of getting confused over there. This is all positive. Now this is A negative. It can give to, of course it can give to A. See friends, positive, negative can give to positive and negative. Suppose I am positive, I can give blood to positive, at the same time I can give blood to negative also. You are getting the point. So now suppose example, my blood group is A negative. I can give to A negative, A positive also. Anyway I can give to AB positive also, AB negative also. But I want to receive. I want to receive means antigens has to be complement. You are getting the point. Then A negative can receive from A negative and O negative. You are getting the point. Now the blood group of mine is O negative. I am O. I am what? Universal donor. So I can give to anyone. But when I want to receive, that's why they will say na, O negative is the most rarest blood group. Correct na? Very less people having the O negative. O positive, many people are there, universal donors. Correct na? But O can receive only from O negative. No, don't get confused. B minus. B negative can give to B positive also, B negative also. AB positive also, AB negative. So I am a B negative, I can receive only from B and O negative. Correct na? Don't get confused over there. Same goes with AB. AB negative means I can give to AB blood group. I can give to AB positive, AB negative. Then I can receive from, my blood group is AB negative. I have to receive from all negatives. I can receive from A negative, B negative, O negative and AB negative. Now it seems to be fine. If I turn the slide, you will forget. Okay. So the point is that we have to remember them very clearly. Remembering is more important. Okay. Now understand. Now we have to read something different blood group other than ABO blood group, that is called as RH blood group. Okay, please be patient, we'll complete this. Okay, the RH blood group is so named because the antigen was discovered in the blood of rhesus monkey. Okay, however, a blood group having A antigen, B antigen, similarly blood group is also having RH. So a person whose RBC having RH antigen, we called RH plus group. Who do not have RH antigen on their RBC, we called RH negative. Correct na? But interestingly, if my blood group is A, my blood group obviously makes antibody which are anti-B. If my blood group is B, I will make the antibodies of anti-A. But the question is that my blood is making the antibodies. But the persons whose blood group is RH, their plasma do not produce any antibodies. You are getting the point correctly. Their blood plasma do not produce any antibodies. Normally, blood plasma does not contain anti-RH antibodies. Now, when antibodies will be produced? Antibodies will be produced only during the transfusion. Okay, if RH negative person receives an RH plus blood transfusion, the immune system start to make anti-RH antibodies that will remain in the blood. See friends, it's very simple example. My blood group is RH negative. Correct na? So when my blood group is RH negative, I have taken the blood from RH positive. Originally I should not take. Correct na? So but when I take that, my body anyway do not have antibodies. Correct na? So RH positive is, can I say that it is a foreign antigen? Then my body recognize and start making antibodies. Before antibodies production, only transfusion is over. Because antibody production will take minimum 4 to 7 days. So which means during the first transfusion, no problem. You are getting the point. Second time also same is happening. That time what happened? Mine is RH negative, I am taking from RH positive. Which means inside my body already antibodies are ready. Already ready. Then they will go and destruct RBC. Then RBC breakdown, hemoglobin releases, kidney failure occur. Okay. When this condition is possible, this condition is possible only in the pregnancy. We'll discuss with a good example. Focus all of you. Don't forget, persons with, see, there is no way of asking extra question. That's why I'm reading line by line. Persons with RH called as RH positive who do not have RH negative. Unlike ABO blood group system, natural antibodies to RH antigens are not present in the blood. Means no natural antibodies, but antibodies can be produced upon exposure. Now look into this line. If RH negative person is exposed to RH positive, which means RH positive is foreign particle for the RH negative. Then my body pathogen enter or antigen enter, my body start making antibodies. But it takes time. It takes 4 to 7 days. So meanwhile the transfusion is also over. So first time no reaction occur. So hence along with the ABO blood grouping, RH blood group is also matched before transfusion. Now formation of RH protein, this is important for the exam where we can expect the questions in the exam. Formation of RH protein is controlled by a gene named as dominant gene called as D. Now friends, you only tell me RH protein controlled by a gene called as D. So can I say it is what? Homozygous, dominant. This is dominant. Now it can code for positive or negative. Obviously I am using what? Dominant. Dominant obviously code for what? Positive. If it is homozygous recessive, code for negative. Correct or not? So thus DD homozygous and DD heterozygous both code for dominant, that is RH positive. Homozygous recessive individuals with the DD genotype is RH negative. Correct na? But friends, when you observe phenotypically, what do you mean by the word phenotypically? When you see the blood, you can see RH positive is also looking same, RH negative also looking same, but genotypically they are different. So this kind of statements comes in the exam. They'll give a simple statement RH positive and negative phenotypically, genotypically normal. Is it right or wrong? No. Wrong statement. Phenotypically normal, genotypically different. Now tell me when the problem occurs. Problem occur only, see, now you tell me RH negative, RH negative. Any problem?
No.
Means donor recipient. Any problem? No. RH positive, RH positive. Any problem?
No.
Because blood groups has to match. Now matched, no problem. So now tell me this problem occurs or not?
Yes.
Obviously occurs. Similarly, problems due to RH blood group arises during transfusion and pregnancy. Now look into this. There is no point of getting confused. It is easy to understand. The first blood transfusion of RH positive blood to the persons with
RH negative.
causes no harm. Why?
Because antibodies are
Production takes lot of time. The RH negative person develop antibody or anti-RH factors in his or blood after exposure to over a period of time. That's the reason it takes time. That's why there is a no harm. Now in my body, obviously I have produced RH antibodies. So second blood transfusion, RH positive blood to the RH negative person. Then in my body, what antibodies are there? Antibodies against RH positive. What it will do? It destruct red blood cell. Then there is a release of hemoglobin. Correct na? Now, when this condition is possible, friends, this condition is possible in the pregnancy. Okay, this is the question more than asked 3 to 4 times in the exam. So, you all know very well, during the delivery time, it is possible placental circulation, maternal circulation can combine. Correct na? There is a great possibility that it makes, not a greater quantity, very small quantity. At birth, small quantity of the fetal blood combines with the placental. Correct na? Now just try to understand. Mother is RH negative. Baby is RH positive. Now during the first time, placental and maternal circulation got combined. So little blood got transfused from baby to mother. Correct na? Then mother start producing and not RH. You should always use anti-RH. Antibodies again this RH positive antigen. Correct or not? But first time there is no harm. Delivery occurs successfully. Correct na? When the problem occurs, second time also if the baby having the same RH positive, that time the problem come. What happened? Now again during the time of delivery, what will combine? The maternal circulation and fetal circulation combined. Then automatically already inside the mother's body antibodies are ready. Then anti-RH attack on the fetus. Correct na? Then fetal RBC get destructed. This condition is called as hemolytic disease of newborn, widely called as erythroblastosis fetalis. Give me why we call it this, friends? Erythroblastosis, breaking down in the fetus. That's what we call erythroblastosis fetalis or hemolytic disease of newborn. When it is happening only during the second pregnancy time. Correct na? So what is important, friends? Important is blood groups has to match. So quickly, a special case of RH incompatibility has been observed between RH negative blood of a pregnant mother and RH positive. Now very interesting line here, look into this. If father is
RH positive.
mother is
RH negative.
now tell me, friends, what is the possibility of fetus blood group? Positive. Because it is dominant. Because either homozygous dominant or heterozygous. So 90% of the cases positive only. Then obviously it cause what? Serious condition in the during the pregnancy time. The first because this is the statement which we can get. The first pregnancy of the RH negative mother with RH positive fetus is normal, provided the mother has not exposed any RH positive blood through the transfusion previously. So no antibodies. Hence no worries at all. The only problem occur second time. Okay. So it is very easy to understand. Now we understand incompatibility during the pregnancy. Okay. Now friends, if you get a very tough question, you will get from here. This is the last and final one. Then rest everything is easy. Blood transfusion. Just now we understand the wrong blood transfusion with respect to the RH group. So you have to understand very clearly what generally happens once I receive a wrong blood group. What exactly happens? So now tell me, when I use the word transfusion, what generally I will take? Sometimes we will say, a person met a severe accident. Okay, that time we will transfuse whole blood. Suppose example, now you think like this, a person having anemia, then I have to take only RBC. Is there any necessity of WBC? No. So depending on the condition, sometimes we will take whole blood. Sometimes we will take only plasma. So a transfusion is the transfer of whole blood or blood component. Either only red blood cells or only plasma. Suppose a person require abundant amount of albumin, then no point of taking RBC. We will take only plasma. Either we will inject the blood directly into the bloodstream, or some genetic diseases we directly introduce into the bone marrow. Correct na? Bone marrow is the site where all the RBC production occur. Now see here, very important. There are multiple reasons why we do the transfusion. A transfusion is most often given to elevate anemia. Correct na? To increase blood volume. After severe, what is hemorrhage, friends? I told you, internal bleeding. Correct na? Don't forget, okay? Internal bleeding, that is called hemorrhage. That time only we do the transfusion. So friends, now don't confuse, I'll explain slowly. I want you to understand the concepts correctly. In an incompatible, what is called incompatible? Wrong. In an incompatible blood transfusion, antibodies in the plasma bind to the on the which cause agglutination or coupling of the RBC. Agglutination is an antigen-antibody response in which RBC become cross-linked to one another. So now, can you explain me this line very correctly? What it is saying? There is a wrong blood transfusion occurred. So I am a recipient, correct na? In my body, how much blood is there? What is the average blood? 5 to 6 liters. So I am a recipient. In my body, I am having 5 to 6 liters of blood. How much blood I will take generally? 300 ml. Maximum we will take only 300 ml. So slowly think and answer. In my body, more antibodies are there. In the donor blood, more antibodies are there. In recipient, I have 5 to 6 liters of blood. So in my body contain more antibodies. From donor, I am taking hardly how many? 300 antibodies. 300 ml of blood. Less antibodies. So don't get confused here. This is the blood group. What is the blood group? A. Okay. Blood group is A. Now what is the antigen? Anti? B. Correct na? Antigen ma, antigen. Antigen is A. Is B. Correct na? Can I take the blood from B? No. Correct na? I can only take from A or O. Good. I cannot take from B. So if I am taking from the B, it is undoubtedly incompatible or the wrong blood transfusion. Now tell me on the B, what kind of antigen is present? B. And? Wonderful, wonderful. Anti-A. Correct na? Now it is crystal clear. A is recipient. B is donor. Now you tell me slowly, whose antibodies are more now? Anti-B or anti-A? Anti-B. Anti-B. Always recipient having more blood, more antibodies. Correct na? Now the blood transfusion occur between these two. Now anti-B can easily attack on antigen B present on the donor. Correct na? Now anti-B can easily attack on this B. Then what happens? This RBC will be destructed. Hemoglobin will be released. This is called serious reaction. When it is not serious, now you think like this. This is donor. Donor having what antibody? Anti-A. So it can also act on A antigen present on the recipient. Correct? Then you have to answer why it is not serious. Because less antibodies. So what? Less antibodies can easily diluted in the blood plasma of recipient. We have a lot of plasma, all will be diluted out. They won't show any reaction. So question is very clear in the exam. Remember, when recipient antibodies attack on the donor antigen, that time only reaction is severe. When the reaction is less severe, donor antibodies when attack on the recipient antigen, because they are less in number, they can easily get diluted in the blood plasma. Correct, na? So those are about blood groups and what we can call blood transfusion. Here in case of human physiology we get direct question. Sometimes the blood groups they link with the genetics, and there they will raise the questions. Okay, I will explain once the madam explain it. Okay, now plant physiology is going on, right? Yes. Okay, so whatever I have not thought that like factors affecting photosynthesis, discoveries of the photosynthesis, read compulsory. Okay, then it become a good revision to you. Now the next one is okay, we'll solve some questions. I don't know how many questions are there, because as I said, most of the questions will appear from ECG. Then cardiac system, from blood groups there are very less number of questions. Please revise, okay, maximum by October ending or November first week we will complete and we'll do vigorous revision. You have to become super duper experts. One second, which year it is? It is not kept. I think 2022 it is. Slow. Spend 1 minute. I forgot to say this horseshoe shaped or kidney shaped. Now what's the question? Correct. B and
C.
So what is the answer?
B.
Why this is wrong?
Abundant.
So which are more abundant? Correct, na? So they recruit histamine, serotonin, heparin and PAF. Remember, next time if they ask this, you should not think platelet activated factor. Since histamine, heparin is there, they are involved in the inflammatory. Correct, na? Now which is having kidney shaped? I think neutrophils only. No, eosinophils. Horseshoe shaped or kidney shaped nucleus. Okay, or thrombocytes. Okay, we'll see this. See this compulsory. I forgot it. And basophils are what sites? Granulocytes. Correct, now? The answer is B and C. Okay. Now I want you to answer this question. See, whichever not there in NCERT, whichever I explained. See, look into that correctly. Always eliminate, friends. This much fast.
C.
Okay, others?
C.
What is yours? C. Trust my words, if you answer this much fast, you will make all mistakes in the exam. Okay, always have patience. What is glycogen? Storage product of glucose. Where the glycogen is getting stored? Liver, muscle, heart, in the brain. So what is the name of the hormone that is involved in the breakdown of the glycogen to glucose? Very good. What is it? Not glucocorticoid, glucagon. Correct, na? In the liver, in the muscle. That is the question we have seen in the endocrine system. Epinephrine. Correct, na? So you are not reading. Okay. Epinephrine. Correct, na? What is globulin, friends? See, this is the statement which is not there. What we discussed, globulins involved in the transport also and immune system functions also. So globulin is acting like a antibody. Correct, na? And where it is present in the plasma, released from liver cells, hepatocytes and steroids. Example, all these sex hormones and all the androgen, estrogen, progesterone, testosterone, cortisol, all these are all steroid hormones. Correct, na? And what is the next one? Thrombin. What is thrombin? What is thrombin? It is there biocatalyst you are saying. Very good. It is thrombin function. Ultimate function is what? We will discuss that. Conversion of soluble fibrinogen into insoluble fibrin. You remember we use the word blood clot. We'll see in the next slide. This is a RBC, right? See, blood clot means we have a lot of threads. Okay, very good. Thin mesh network like structure, which is fibrins. That fibrin can be formed from fibrinogen. This is soluble, this is insoluble. That's why we can form the threads. Okay, so it is what? Biocatalyst. Now tell me why we use the word biocatalyst. That is my question. It is an enzyme. Correct, na? Thrombin function is what, dear? Conversion of soluble fibrinogen to so it is an enzyme. That's why they use the word biocatalyst. Okay, it's not necessary they have to give enzyme, right? Okay, so it is a biocatalyst. Okay, that's the important. You have to understand it correctly. No. This is from heart. Okay, we haven't discussed. No, I'm just seeing. Try it. We haven't discussed, but you can see. Just now we discussed that only it came, but it's okay, fine. Try it. Option C. Okay. Why? It is called as what? It is called as fibrins. What is called coagulum? Clotting. The clotting formed is network of threads called as spleen is the graveyard of erythrocyte, whereas which cell? Macrophages. Very, very good. Okay. B, yeah? Correct, AB blood group, what kind of antigens it is having? Antigen A also, antigen B also. Very good. We haven't studied. Correct, na? See how many times they have asked the same question in a repeated way. Okay. Anyway, this is again ECG, ECG, ECG. This is heart, ECG. See how many questions are again heart. So your favorite word, Sanjana? See, again you all people, again I want you to answer correctly.
D, sir.
D, yeah?
A2, D3, C1.
Correct, na? So, globulin is always involved in what function?
Defense mechanism.
So, in the NCERT it always written that it is involved in the transport. But alpha, beta, in that especially gamma globulin involved in the defense mechanism. Okay. Now see, this is the question I am waiting for, which is not there in the NCERT. Is appropriate.
D is oxygen.
See, all human RBC are enucleated. That is right. Correct, na? So, you know, right, ma? Eukaryotic cell, which organelle is most occupied in terms of a space. Suppose example, you take a plant cell. This is the plant cell, contains cell wall and cell membrane. Correct, na? Inside the plant cell, which is the largest organelle, because it is the one occupying a lot of space. Similarly, if you take a eukaryotic cell, which is occupying the large space, which is present centrally, nucleus. Nucleus is occupying more space. RBC do not have nucleus only. Then what about the remaining space? Lot of space is there. Lumen, hollow lumen, complete internal space available for the transport of oxygen. Correct, na? See, they have asked a very good question. Most appropriate means most suitable or eligible answer you have to remember. Okay. Now they do not reproduce. How come it is wrong? You remember, no ma? RBC division won't occur, dear. Correct? Now what we discussed, RBC will die. I just explained the entire concept in the last class to make you understand this kind of lines. You remember my words? First RBC destructed, giving rise to globin and heme. Very good. Heme contain iron part and what is this non-iron part? Biliverdin, bilirubin. Iron is Fe3 plus. Remember, what happened to this globin? Break down into amino acid and further converted into proteins. You remember this Fe3 plus enter into the liver with the help of ferritin. You remember? Finally it enter into where? Bone marrow. In the bone marrow it is combined with erythropoietin. Correct na? And then it is produce RBC. So it never reproduce. Getting the point? Just to explain this I told you. Okay. They are not completely somatic cells. Okay. They do not metabolize, they metabolize. Okay. See, I told you, right, it will show the aerobic respiration. Sorry, anaerobic respiration. So they perform the metabolism. Okay, don't confuse with the statement. So the true and most appropriate statement will be the option D. Okay. Anyway, remaining all our cardiovascular system, we will read in the next slide. Something is this? Okay. Huh? Correct na? Which are thrombocytes. See, people who used to get 360 out of 360. See, they are getting 360 out of 360 because of simple questions. Okay. Now no one is not getting 360 out of 360 because the level of questions are increased. Okay. What is this? Lacking of clotting factors. Very, very good. Okay. Remaining we will continue in the next class. Tomorrow we will discuss the coagulation of the blood and we will discuss most important cardiovascular system. All right. Okay. So now I don't think you have this week exam, you have next week, next Friday. So next Friday we'll complete this also, and you wanna write only one or you can write two. You all said that breathing exchange of gases is very easy, no? Okay. No problem. In that case you write both, okay? But if you are feeling difficulty, we will go with one. Okay? Done. Read the biomolecules.